All right, hello everybody. We're going to talk about meiosis today. We've already talked about mitosis previously. Remember that mitosis is cell division, where we're basically cloning cells. We end up with two cells that are identical to each other, and they are identical to the parent cell. Now we're going to talk about meiosis, which is the formation of gametes, sex cells. All right, let's get started. So first of all, meiosis is often referred to as reductive eukaryotic cell division, and that sounds fancy. It's referring to the reduction of the number of chromosomes. So mitosis is non-reductive eukaryotic cell division, and that's because it didn't reduce the number of chromosomes. So if you had a human somatic, that's a body cell, if you had a human somatic cell that had 46 chromosomes in it, and the cell went through mitosis, each of the daughter cells have 46 chromosomes. So we have not reduced the number. So it's non-reductive. In meiosis, if we have a human cell that starts with 46, and that's called a germ cell, if it's the first cell that's going to divide into the gametes, the first cell has 46 chromosomes, and then by the time you're all done, each of the four cells has 23 chromosomes. All right. And then just a quick vocabulary word that you may not have heard before, the word zygote. The word zygote is when sperm and egg come together. Okay, moving on. Chromosomes in a diploid cell. So remember that a diploid cell has pairs of all the chromosomes. So humans have 23 pairs. That's our diploid number. Our diploid number is 46. The haploid number is half the DNA, and that's where you have one of each of the chromosomes. And so that's called the haploid number, and that's what's in gametes. Take a look at the diagram here because this blows people away every time. It's very, very confusing. What you're looking at is something that's called a karyotype. It's a photograph of someone's DNA. And it's, if you look, it says 1, 2, 3, 4, 5, 6 through 22. So there's 22 pairs of chromosomes. Those are chromosomes. And then the last pair is an X and a Y, which means that this is a biological male. So each of those chromosomes, some of them are standing kind of crooked, some of them are standing upright, some of them are leaned over. That's because they have the consistency of wet noodles, and so they don't always stand upright. That's fine. But what you're looking at are pairs. And so one on the left comes from mom and the one on the right comes from dad, or vice versa. But they're the pairs of the chromosomes. And in this photograph, each one of the ones that are standing there, each one of those are bivalent. So take a look at the cartoon on the left. We're looking at two that happen to be leaning sideways. This is not an X. This is not a bivalent chromosome. It is two bivalent chromosomes. There's a bivalent chromosome on the left and there's a bivalent chromosome on the right. So take a look at that diagram and see that there's two parts of each side, and those are the sister chromatids. So right now, there are actually 4 little chromosomes put together. There are 2 identical in a bivalent chromosome and then 2 homologous ones in the next bivalent chromosome. Hope that made sense. And remember that we call the 2 pairs homologous chromosomes, homo meaning same. So they both, for example, I always use this example, they might both carry a gene for the shape of your chin. But dads might say you have a dimple chin and moms might say you have a smooth chin. The gene for the shape of the chin, that's called a gene. And then whether it's dimple or whether it's smooth is an allele. But the two together, this chromosome and this chromosome, those are the homologous partners. And they're sometimes abbreviated as homologs. So you might see that in a test question. All right, moving on. So then, taking a look at this picture, you see that all of them are bivalent, so they have 2 copies of each other, but they have partners. This is a stage of mitosis. This is prophase. So I know it's not Prophase One of meiosis. We'll get to that. So if you're reviewing this video, it'll make sense. If you're seeing it for the first time, you'll be like, what is she talking about? I know it's not Prophase One of meiosis because the homologous partners aren't standing with each other. So they don't stand in a line, they just partner up. It's called synapsis when they partner up so that they can cross over. If you're like, what is she talking about? It's going to come up in just a second. But this is a picture of prophase of mitosis, and you can see each of the chromosomes. All right, let me get my picture in a better space here. Let's go, I don't know, right here. That works for me. Nope, it didn't stay. Will it stay? Nope, it doesn't want to stay there, whatever. Okay, so it says meiosis looks a lot like mitosis. Instead of the phrase IPMAT, interphase, and then prophase, metaphase, anaphase, telophase, remember that interphase isn't actually one of the stages. That's its sort of normal stage where it's doing its thing. So really really mitosis starts with prophase. So prophase, metaphase, anaphase, telophase. For meiosis, look on the right side. Let's see, I got to point to it. I'm trying to point. There we go. This picture. My image is backwards. I never get that. So this picture that you're seeing along this side of the screen, that's meiosis, and it's got more stages, but the stages are really familiar. Instead of IPMAT, it is PMAT. We have meiosis 1 and we have meiosis 2. So it says the first half of meiosis is denoted with the Roman numeral 1. The chromosomes cross over. That happens in prophase 1. I can't point to it. There we go. Prophase 1, the chromosomes cross over to create new genetic combinations. The chromosomes line up single file. That's in metaphase 1 there. I'm sorry, I didn't mean single file. They line up side by side. They line up single file in mitosis metaphase, but in metaphase 1 of meiosis, they line up with their homologous partners. And then the cell divides, and that's where the chromosome number gets cut in half. The second half of meiosis is denoted with the Roman numeral 2, and it's identical to mitosis, but the chromosome number is halved already. So we have haploid cells already. It'll all start to make sense as we go. So here's a closer look at meiosis 1. So we're going to move across the screen. Actually, it's this way there. We're going to move across the screen this time. So you've got prophase 1, metaphase 1, anaphase 1, and telophase 1. So let's take a deeper dive into prophase 1. Let's get my picture out of the way here. Okay, so for prophase 1, the homologous pairs, they find each other, and that's called synapsis. And then they form structures that are called tetrads. The word tetrad means 4, or the prefix tetra means 4, and that's because these are two bivalent chromosomes that come together and they cross over, and where they cross over is called the chiasma. And now they're going to trade sections of DNA, and if I could trade fingertips, I would show you, but I can't. So they change sections of DNA. So what was on mom's chromosome and dad's chromosome, now they trade, and there's a little bit of mom's chromosome on dad's and vice versa. So this is called crossing over, and it creates genetically unique chromosomes that have never existed in the history of the world before, which is kind of cool, and it adds to our diversity. So crossing over, every gamete has a unique sequence of DNA in each chromosome. So take a look at the daughter cells down at the bottom. Those are the gametes. Those are the sperm, egg, or pollen. They're haploid. They have half the DNA. And you can see that the one that's just blue, it got, let's say, mom's DNA. It doesn't matter. It got a chromosome that's just like mom's. But look at the one right next to it. It's got blue and red tips. That's from crossing over. And so this is a combination between mom's chromosome and dad's chromosome. So if that, let's say that's an egg, if that egg gets fertilized by a sperm, this child is going to have combinations of the two grandparents, not just one chromosome or the other from them. Okay, and then the word recombination gets used all the time in genetics, and it means recombining DNA from two different sources. So that could be crossing over. We get recombination between what are going to be the baby's grandparents. That's crossing over. You could have that in a genetic engineering experiment where a scientist takes DNA from a jellyfish and adds it to a goldfish, and now you've got recombinant DNA. But that's what the word means, and it's adding to genetic diversity. Okay, so we're still on this image. It's the same image of meiosis 1. We looked at prophase 1. Now we're going to scoot over to metaphase 1. And in metaphase 1, they line up with their homologous partners. Now take a look. Oh, they haven't shown it very well, but they show that some have crossed over. I'm not wild about this photo. It's not our diagram, but they're lining up side by side. So that's different than metaphase of mitosis, where they line up single file. Back to the picture, we're on to anaphase. Now this picture here really clearly shows you that the chromosomes have crossed over, and that notice that there's like a blue chromosome with red tips. So you've got these genetically unusual chromosomes. So it says the homologs are separated, but they remain bivalent. When the cell divides in telophase 1, it's going to divide right between them. When the cell divides in telophase 1, each chromosome is still bivalent. So the cell divides a second time to distribute the extra copies of the chromosomes. So when the cell goes through telophase, we're going to get down to the half number. We're going to have haploid cells now. But each of these haploid cells has still got bivalent chromosomes. So we want to separate the bivalent extra DNA. So it says, notice that each red chromosome has some blue, and that's due to crossing over. All right, and then telophase is what makes them haploid. And now we'll go on to the second half of meiosis. So this is Meiosis II. So the cells have divided, and now we have two cells that are both going through prophase 2. Notice that they are still bivalent, but the number is in half. We started with 6 chromosomes. I'll remind you, we started with 6 chromosomes. Now each of the cells has only 3 chromosomes in it, but they're bivalent. So in prophase, the usual things, you start to see the chromosomes. In metaphase, this time they line up single file, just like in mitosis. Why don't they line up with their partners? Because their partner's in the other cell. They can't line up with their partners. So they line up single file. They get ripped apart in anaphase. So now these monovalent or sister chromatids, but the monovalent chromosomes move to opposite sides of the cell. And then the cell divides. And so now you should end up with 4 gametes. They are haploid cells, and they would be sperm, egg, or pollen. Just a couple quick FYIs. It's called spermatogenesis when it happens in a guy and produces sperm. It is oogenesis when it happens in a female. And when it happens in a female, it's a little bit interesting because only one of the 4 eggs gets all of the cytoplasm and all of the organelles. So it's like a huge one, and then 3 itty bitty little ones, and the 3 itty little bitty ones, they go away. And so the female, when her body has gone through meiosis, she gets one gigantic egg out of it. When a male goes through spermatogenesis, they get 4 teeny tiny little sperm out of it. And the sperm don't carry any of the organelles. So the sperm are basically packets of DNA with tails on them. That's all they have in them. So the organelles come from the female, and that's going to become important later because you're going to inherit your DNA that's in your mitochondria. Remember that mitochondria have DNA because they used to be bacteria. They have their own DNA. You're going to inherit that through the female, or the matrilineally, I think is how they pronounce it, through the mother's side of the family. You're going to inherit all the mitochondrial DNA. Okay, so some more about chromosomes. Sex chromosomes are the X and the Y, but all the other chromosomes are called autosomes, and that's definitely a word you need to know. It seems like, why do I need to know a different word for chromosome? But the sex chromosomes are super different because, at least in humans, and we'll talk about some variations, humans, the females get 2 copies of it, and males, they only get 2 copies of the X, and males, they only get 1 copy of the X and 1 copy of the Y. And so that's really important in the differences and how we inherit, how our sex is formed, but also how we inherit different genetic traits. So the autosomes were the same between male and female. So a male gets 2 copies of chromosome 1 and a female gets 2 copies of chromosome 1. So there's no difference between that. And then just a quick FYI, remember that the idea of gender, how you feel about yourself, how society sees your roles, are not necessarily determined by biology. So that's a gray area that we're still learning a lot about. Okay, sexual reproduction. The purpose of sexual reproduction, I'm kind of in the way here, is to increase variation. Asexually reproducing organisms like bacteria, for example, they just clone themselves. So they only generate variation through mutations and through something called horizontal gene transfer. Bacteria have the ability to pick up DNA from another bacteria, plasmids, tiny little circular pieces of DNA. They can pick that up from their neighbors, and then they can pick, I'm sorry, I'm daydreaming here. They can pick it up from their neighbors, and then they can change a little bit instead of sexually reproducing organisms, which combine the DNA, half from one parent, half from the other, and then you've got this totally unique offspring. So sexual reproduction increases variation. All right, let's talk about a few things that are super important in meiosis. The first one is independent assortment. Independent assortment refers to the fact that all of the chromosomes, the homologous pairs, they line up independently of each other. So each pair lines up independently. So pair number one, you might have moms on one side and dads on this side. Pair number two, you might have moms on the same side and dads on the same side. But pair number three, they might be reversed. You might have moms on this side and dads on that side. So it's completely random which order the pair lines up on. And so that means when the cell divides, which chromosomes go into which eggs or sperm or gametes is completely random. And that, because we have 23 chromosomes, that is an enormous number. It's 2 to the 23rd possible combinations of chromosomes in our eggs and sperm. And we're not even talking about the diversity that's added by crossing over. All we're doing is looking at a whole chromosome and whether it goes to the right cell or the left cell. So that's more than 8 million possible genetic combinations in any given human sperm or human egg. You bring them together, an egg and a sperm, and it's something like, I don't remember off the top of my head, it's like 70 trillion combinations in one baby. Like the odds of having, maybe it's 70 billion. I don't remember. You guys can do the math. But anyway, it's crazy. All right, so just a quick reminder what the word recombinant DNA means. It's anytime there's new sources or new combinations of DNA. So crossing over adds to recombinant or causes us to be genetically recombined. Mutations cause new DNA, new sources of DNA, and then random fertilization. 70 trillion, I was right. Random fertilization, there are around 70 trillion possible combinations of unique chromosomes in the zygote, in a human zygote, for example. So you truly are unique. Okay, and then just a quick reminder again about why sex. Asexual means without sex. And so their bacteria reproduce asexually. They just clone themselves. The advantage is that it's super fast. So bacteria are able to reproduce every 20 minutes. And that's partly because they're small also, but they don't need to find a mate. So reproduction happens much faster. I will want you to realize, though, that there are many organisms beyond just bacteria that can reproduce asexually. Strawberries, for example, have the ability to release a runner, and there's many plants and trees that can do this. They have the ability to release a runner, so it looks like a stem that's growing along the ground, and then that stem can grow roots, and a new plant can grow from it, and that plant is a clone of the original parent plant. Now, the parent plant also has flowers. So if a bumblebee, a pollinator, happens to come by and get the pollen and fertilizes another strawberry plant somewhere else, then that's sexual reproduction, and yay, that increases genetic diversity for the strawberry. But if no bee comes by, the strawberry plant doesn't have to wait for a pollinator in order to reproduce. It has the option to clone itself, or it can reproduce sexually. So it has both, which is kind of superior. All right, so the disadvantages to asexual reproduction, to cloning, is there's a lack of variation. So just imagine if all humans were clones, and some of us really, we'll use COVID as an example. If some of us died, which we did, if some of us died from COVID, then we would all be equally susceptible to COVID, and this could devastate the human population. But because humans are so diverse, the way in which we responded to COVID was so different. Some people died, but some people didn't even have symptoms. They were asymptomatic. And that's because of our genetic diversity. So it increases the chances, being genetically diverse increases the chances that if something happens in the environment, a new disease, a new weather pattern, so like global warming or global cooling, new predators, new food sources, or the loss of food sources. Any of those environmental factors, if they happen, if you have a diverse species, then you're more likely to have some individuals in that species who can handle that new change, because you're not all exactly the same. All right. So it says for asexual reproduction, if the environment changes, there may be no individuals with the needed traits, so the population might go extinct. So sexual reproduction, the advantage is that it adds genetic variation and increases the likelihood that someone will be able to handle new changes in the environment. The disadvantage is that it takes time and energy to locate a mate. So reproduction is slower. It's much harder. I love my little poop beetles. What are they called? Dung beetles. There we go. Trying to find a little mate and talking over a poop ball and whatever. Okay, so they have to find a partner, and that's a lot more complicated than just cloning themselves like a strawberry plant or a bacteria. Okay, this is the last slide, everybody, just comparing mitosis and meiosis. Remember, mitosis produces two identical cells to the original parent cell, which would be a somatic cell. And then meiosis produces four gametes that are haploid. They are completely unique. And the original cell, the first cell, is called a germ cell. That's it. Have a great rest of your day.